So in the last tutorial segment, we learned how to determine the correct gene order for our map. And just to review our objective, the three-point linkage mapping problems are a way to use data collected from the outcome of a mapping test cross to determine the order of 3 genes on a chromosome and the relative distances between the genes. So we've already determined the order. The next step is to determine the relative distances between the genes. So here's our example problem again, and you remember that we have determined that the W gene should be in the middle of the actual gene sequence on the actual chromosome. And we did that by comparing our parental groups with our double crossover groups. So you remember that W was in the middle, so what I would suggest you do is look at your parentals and rewrite the parental genotype in the right configuration. So big S, big W, little y, so putting this back in the correct order. And then on the other chromosome, on the other homolog, little s, little w, big Y. So now you have the 2 parental chromosomes, and remember this is this parent here. We're laying out the configuration that triple heterozygous parent must have had. So some of the offspring got this chromosome completely intact without any crossover, so they got big S, big W, little y, and that's our 805 group. And some got little s, little w, big Y, and that's our 813 group, and those are what we call the parental allele combinations. The rest of the groups here have chromosomes that during meiosis in this parent's cells got crossovers, and so the chromosomes they have represent crossover chromosomes between these 2 homologs. So once you write out your parental genotype, you want to keep it, and I would usually write it on the side here and keep it for reference for the rest of the problem. So I've just moved it over here. Big S, big W, little y, little s, little w, big Y. Okay? So now we need to figure out the distance between S and W. We're going to call that segment 1 of our map, and then we'll figure out the distance between W and Y, and we'll call that segment 2. Remember that recombination frequency can be used as a measure of genetic distance. So if we can figure out how many of these offspring got a chromosome that had a crossover in segment 1, then we can figure out our genetic distance for segment 1. So you're imagining a crossover in segment 1 during meiosis. So use your parental configuration to identify any progeny groups that might have a chromosome that would result from a segment 1 crossover. And you'll be looking then for anything with a big S and a little w, or a little s and a big W. Those will all be included in our calculation. So when you look through the data, you see, you have to ignore the Y, but you see this one has big S and big W, so that's not something that we're looking for. Then you see big S with little w, that is. This one down here, big S with little w, that is. Little s with big W here, and then this one, little s with big W here. So you identify your groups that have these allele combinations, because that means those chromosomes are the result of a segment 1 crossover. So you're going to count 123 plus 8 plus 10 plus 118. Divide by the total, 2,000. You'll get 0.1295, but we really want it in the percentage form, 12.95%, and the genetic distance then between S and W, and it could be a big S or a little s, it could be a big W or a little w, depending on which individual's chromosomes we're looking at, but the distance is consistent. It's always 12.95 map units between those genes, no matter what alleles are at each of those gene positions. Now, to do a segment 2, you're going to imagine W, if this crossed over here, you'd have big W with big Y, or little w with little y, so you look through your data again and you pull out the data that fits those patterns. So here we have a big Y and a big W, so 60. Here's a little y and a little w, 8. big Y, big W10, little y, little w63. And so when you add up those groups, you get 0.0705 or 7.05%. So that's the distance. 7.05 map units will be the distance between any allele of W and any allele of Y on any of these plant chromosomes. One thing you'll notice in the calculation is that you use the DCO groups, the double crossover progeny groups are counted in the segment 1 calculation and the segment 2 calculation. Because by definition they had a double crossover. They had a crossover in segment 1 and a crossover in segment 2. The other ones will just have a single crossover. And then the ones that are never used in the numerator are the parental groups. So the 805 and the 813, those parental groups should never be included in the numerator of your recombination frequency calculation, because a parental chromosome didn't have any crossover. So it's not recombinant. So it's excluded from the numerator. So if there's a point where you think that you should include a parental number in your numerator, it means that you either didn't figure out your gene order correctly, or you didn't lay out your parental configuration correctly, or something's wrong. So that can be a clue to you to go back and double check. So then we have the map that we now can draw. I would usually draw a line with little hatch marks, and then you put the gene names, the gene abbreviations, or whatever you want here, S, W, and Y. You can put lowercase letters, you could put uppercase letters. This is generic, meaning it represents any chromosome. So any version of S, big or little, any version of W, big or little, any version of Y, big or little. But the generic chromosome will always have 12.5 or 12.95 map units between S and W, and 7.05 map units between W and Y. So the distances are always consistent, and the order is always consistent. W's in the middle. Now you could rotate this whole image 180 degrees. You could have Y over here on the far left, but then you need to have 7.05 map units to W and then 12.95 map units to S. That would be fine. So just to review, our purpose of the whole process was to use the data collected from a 3-point mapping test cross to determine the order of 3 genes on a chromosome and the relative distances between them. And then you draw the map. So we've accomplished that. Now there's a few more calculations that will be covered in the next tutorial section, but at this point you have created your map.